clearing the malignancy.
In this video, I'm going to be talking about whey protein and how it's related to, or how it can be in context with tumors and nutrient shadow zones. So whey protein is a higher blood amino acid levels, and it does this faster and more intensely than most other protein sources. And why it does that? Because of fast digestion. Whey empties from the stomach quickly, and then rapid amino acid absorption. And then you get a sharp postprandial amino acid surge, especially within the first 30 to 90 minutes. Tumors, especially in nutrient shadow zones, especially after you break a fast, within the first 5 minutes to an hour, that's when tumors can take advantage of any dietary substrate that comes in after digesting. So a very high availability of amino acids like leucine, isoleucine, and valine, and very little is lost to digestion or during digestion. So compared to other proteins, whey has a fast, high peak AA levels. Casein, like in cheese, slow sustained AA release. Means biologically is strong mTOR activation, increased muscle protein synthesis, which could be a good thing for a human, but less effective for long-term amino acid availability unless paired with fat, fiber, or casein. Now, unfortunately, high circulating AAs, especially leucine and glutamine, can also feed highly glycolytic or mTOR addicted cells. Timing and dose does matter more than people think or know. Now, the most pure mTOR activator is whey isolate. 90 to 95% protein, fast absorption, a very high leucine density. Net effect, sharp leucine plus essential amino acid spike, strong mTORC1 activation, or mTORC1, and I have mentioned that in some of my other videos. One is a pharmacologic protein, whey hydrolysate. Okay, so basically it has the highest insulin spike. Now, tumors, especially at the end of a fast, when a fast is broken with, like, let's say glucose or anything that spikes insulin, a tumor can actually sense the spike. So that's like the signal to the tumor to start to feast or to feed on the dietary intake of the human being. Okay, so whey hydrolysate, or WPH for short, small peptides stimulate insulin independently of glucose. Okay, so tumors at the end of a fast can sense the insulin spike from glucose, especially if the insulin is spiked with a high carb meal. But a protein, especially this protein, whey hydrolysate, can spike insulin high as well. So there's a possible chance that that tumors can still sense an insulin spike from protein the same way as a glucose spiking insulin. Okay, so here's where it might benefit the tumor, is that isoleucine and an amino acid as well, even just 1.5 grams can raise insulin sensitization plus mTOR support. Okay, so whey punches insulin without glucose, which is unusual. Also, other insulin-stimulating AAs, leucine, beta cell depolarization, arginine, direct insulin secretion, threonine, incretin enhancement, and lysine, insulin release. Okay, this is important. Okay, tumors upregulate IGF-1 receptors quite often. And now whey does not directly raise IGF-1, but whey, it does increase insulin hepatic GH sensitivity, and then chronic highway chronic excessive intake of whey protein leads to modest IGF-1 elevation. So there's a connection with tumors there. Since tumors, they do upregulate IGF-1 receptors, meaning that any IGF-1 elevation by whey will be sensed and picked up by IGF-1 receptors on tumors. Okay, now here's where tumor nutrient shadow zones come in. So basically, tumors create regions where glucose is scarce, oxygen is low or hypoxia, immune cells starve, and then CAFs, or short for cancer-associated fibroblasts, act as nutrient brokers. Okay, this is where it could be a good idea to include protease fruits such as pineapple, papaya, and kiwis in your diet. Not exactly together with whey, if that's not an option, but the reason is because CAFs, or cancer-associated fibroblasts, they uptake high circulating branch chain amino acids from protein powders. So CAFs preferentially buffer amino acids.
Cancer attacks immune cells with PD-L1, using tunneling nanotubes to steal their power. Green tea activates SCARA5, disabling the nanotube.
Then, through TNTs, or tunneling nanotubes, exosomes, they shuttle AAs toward tumor cores, or the amino acids are shuttled or transferred through the tunneling nanotubes toward tumors. Seems that exosomes, they load up the amino acids into possibly the MVBs. Therefore, tumors don't need a blood supply if CAFs act as amino acid reservoirs. Now, the reason I included pineapple, kiwi, and papaya is because they're basically protease fruits, and not all fruits have the same protease function. But these protease fruits, they do like a cutting, cleaving action. So TNTs, tunneling nanotubes, they have adhesion molecules or adhesion machinery that literally connects one cell to another cell via the TNTs. Like, for example, a CAF cell to a tumor. So the protease fruits cleaves those adhesion molecules, or at least weakens them, so that the TNT is not connecting a CAF to a tumor.
Oh, RASE TNT in big flaming letters. Add audiocomer nanotubes needs RASE. Tunneling nanotubes are out of control.
Therefore, the connection is broken, possibly reducing the transfer of amino acids from a CAF to a tumor. Okay, so another issue is leucine plus insulin spike reinforces CAF sanctuary mode. So insulin, SNAP23 stabilization. Also, mTOR active CAFs, they preserve nutrient-loaded vesicles. Possibly, they might even be loading the amino acids from the protein intake.
Exosomes are tiny vesicles released by cancer cells. These exosomes attack T cells, weakening the immune response. Too many regulatory T cells have mutated into harmful variants. Grapeseed extract reduces exosome production. Protect your immune system.
To the exosomes as well. Also, mTOR active CAFs, they retain Rab5 endosomes. Now that's bad, because it's better to convert from Rab5 to Rab7, because Rab7 is where exosomes and cancer cells, they really can't survive that way. But CAFs, they will retain their Rab5 status, which is not a good thing, because of mTOR. And of course, some of these whey proteins, they do spike mTOR, because mTOR is like it's basically a growth factor, and cancer thrives off of growth. Also, some other good foods to add in to cleave or cut the adhesion molecules on the TNTs, or the tunneling nanotubes, is fermented foods such as sauerkraut and kimchi. Also, possibly asparagus as well. Okay, so immune cells, they lose the competition because they are insulin resistant in the TME, or short for tumor microenvironment, and immune cells cannot access calf shuttled nutrients. Okay, so quick recap. Insulin leucine mTOR signal favors calf-mediated nutrient retention. Use protease fruits to cut the TNT connections because tumors receive amino acids from calves through TNT. So that's it for the video. Like, comment.